Welcome back to TekTalk. This podcast is produced by Teknos, the TJ Science Journal team. TekTalk episodes will be released once a month, with each episode featuring recent scientific news and interviews with TJ students and teachers. Today, we will be talking about something we are all familiar with, stress. Specifically, what implications does stress have on the body? What are today's scientists doing to fix it? What can students do to help? In science news, we'll be talking about research concerning stress and anxiety in mice. This will be followed by an interview with Dr. Burnett, a member of our very own TJ faculty. Dr. Burnett is the teacher of the semester-long courses DNA Science 1 and 2 and one of the two biotechnology research lab directors. Before coming to TJ, she taught as an adjunct professor at Georgetown University and worked on a number of biology-related research projects, primarily on the effect of aging and chronic stress on the risk for cardiovascular diseases. More on her research later.
Today's science news will be about how obese mice lose anxiety when zombie cells exit their brain. Zombie or senescent cells are semi-dormant cells that no longer divide and accumulate in a given area of the body, impairing biological functions. Notably, the presence of senescent cells contributes to anxiety in both humans and mice. In order to further examine how senescent cells impact anxiety levels, researchers have studied the behavior of mice in various environments. Anxious mice tend to avoid open areas, move along outside walls, and perform poorly in navigating mazes. Observing these behaviors, the researchers concluded that the anxious mice had significant accumulation of fat and senescent cells in the region of the brain controlling stress.
Okay, so why and how did you decide to go into the field of biology?
Well, actually, my initial plan was to study chemistry. So I went to college and ended up double majoring in chemistry and biology because I couldn't decide. I really liked chemistry and I liked being in the lab, but I also wanted to have everything that I did be relevant to like human health, which is definitely more the bio field. And so I ended up double majoring in both, and when I got to graduate school, I ended up in a chemistry department because biochemistry was part of the chemistry department at the University of Texas at Austin, and so my PhD is actually in chemistry. But the research that I did was completely biologically related, so I think the main reason why I wanted to do biology was because I wanted to have an impact on human health and well-being.
Cool. So before you became a teacher, what did you primarily research? And what were some of the most memorable projects you worked on?
So before coming to TJ, I was working for the MedStar Health Research Institute, which is downtown at the Washington Hospital Center. And it's part of MedStar Health. So I had a joint adjunct appointment at Georgetown University. And I did a lot of work with people over there, but my main field was in cardiovascular disease. I was studying initially the relationship between infection and cardiovascular disease. There was some interesting data that suggested that if people had been infected with certain viruses or bacteria during their lifetime, that it increased their risk of having heart disease and heart attacks. And so that was initially how I got into the field. But I ended up doing a lot more than just that. We kind of morphed into studying collateral blood vessel development, which is a way of trying to stimulate the growth of new blood vessels to bypass any kind of blockages that may exist. And so we were looking at the role of different types of cells in that process, particularly lymphocytes, macrophages, T cells, CD4 and CD8 positive T cells. And we were looking at the effects of aging on this process. We were looking at the effects of stress on this process. But probably my favorite research topics that I was looking at was the effect of chronic stress on cardiovascular disease and vulnerable plaque development. And some of my favorite mentors were studying stress in the context of obesity and metabolic syndrome. And since obesity and cardiovascular disease kind of go hand in hand, it was a natural way for me to sort of join in what they were doing and complement their research.
Cool. So can you talk a little bit more about like your projects relating to like the chronic stress in mice, like maybe some of like the more interesting little stories? I remember hearing in class that you were talking about I think one time like you had like the female mice with the male mice, and besides that like how the stress affects like the birth and development.
Sure. So I was studying the effects of chronic stress on cardiovascular disease, and we were using a special model of mouse called the ApoE knockout mouse, which has very high levels of cholesterol. And we were stressing them chronically. So the type of stress that we were using was cold water stress, where the mice had to stand in a centimeter of ice water for an hour a day for about 4 weeks. And although that sounds terrible, it's really no different than like having to walk across a college campus in the freezing cold in the winter, or stand at the bus stop or something like that. And it doesn't really hurt the mice, it just is stressful. So that was the model of stress that we were using, and we were looking at the development of atherosclerosis in their aortic sinuses and in their brachiocephalic arteries. And during this study, we actually were in the process of stressing the mice. We'd been stressing them for several weeks, actually. And one of my postdocs was working in the lab, came into my office, and he said, Dr. Burnett, we have a problem. And I said, well, what happened? And he said, well, one of the cages of mice that we have that we've been stressing, there's babies in there. And so what had happened was when the mice were bred in the animal facility, normally the mice are weaned from their mothers at 3 weeks of age, and you separate the males from the females at that time. And so somehow one of the females had accidentally gotten put into a cage with her brothers or males. And so that had ended up resulting in an unexpected pregnancy, which we didn't realize was even happening until the babies were there. And so at that point, I actually got very excited because I said, oh, we should study these mice. They've been stressed for their entire prenatal development, and I wonder what kind of effect that will have on their cardiovascular disease status. And so we let them grow up. We had to do a control study where we had mothers who weren't stressed, and we studied that little small group of mice to see what kind of interesting results we had. And it was actually very interesting. The mice seemed to have worse atherosclerosis. So I ended up writing an entire NIH grant proposal based on this unexpected result that we had not planned, unplanned pregnancy, and got funded by the NIH to do a more complete study looking at the effect of chronic stress on prenatally as well as in adults. So
cool. So like in general, like for research, like between not just mice, but like were there any like challenges? Like what are some of the challenges that you've encountered?
Well, I mean, research as a whole is always challenging. In general, the rule of thumb I like to give is that 90% of what you do doesn't work when you're doing research, and you have to try again and again. And not everything works in the end, but eventually some things do work if you kind of tweak the system, figure out troubleshooting the problems that you're having. So I've had all kinds of challenges from, instruments failing and animals dying, or the animal model that we thought we were receiving wasn't actually what we received, to postdocs losing all the samples that they had spent 4 months trying to generate. I've had all sorts of issues, as well as personnel issues in the lab where there were conflicts between people who were working for me. So in fact, I think actually that's probably the biggest challenge, because research is so difficult and you have to have so much patience to be able to actually be successful at research. It can be kind of depressing. And so I feel that it's very important to have a positive work environment and to support each other and to have a very team-oriented kind of approach. And so I learned early on that if you have conflicts in your lab and people who aren't getting along, it just brings everybody down. And the best way to kind of approach the whole process is to create an environment where people want to help each other and work together and celebrate everybody's successes, because even if your experiment isn't working, if you're helping someone else on their experiment and it is working, then you can feel good about that. So
So why did you decide to like shift away from like the research side, like become a teacher at TJ?
Well, my favorite part of doing research was actually teaching other people. And so I had a lot of experience with summer students in my lab. I had experience with graduate students who were working for me, as well as postdocs and MD fellows who came into the lab, and just being able to teach them how to do an experiment, how to analyze their data, helping them write their papers. That was the most satisfying thing for me. And I was excited when I got my adjunct professor appointment at Georgetown, because I was hoping it would mean that I would have more of an opportunity to teach. But that didn't really work out the way I was hoping. And so I always kind of felt unfulfilled in that area, because I felt like, gosh, I'm helping these people in my lab, but there's only like 8 of them. And I wish I could be helping more people. And so when I ended up making the decision to come here, that was kind of something that had been in the back of my mind for a long time. The real reason I came here was because there was a lot of reorganization going on in my institute, and I was going to have a new boss, either boss A or boss B, and I wasn't really sure that I wanted either one of them for my boss. And funding was getting questionable. My grant was going to be running out soon, and one of my fellows was moving to Miami because her husband had gotten a job there and she'd gotten a postdoc at the University of Miami. Another one was going to do his cardiology fellowship at Tufts. Another of my staff was expecting her second child. And so I thought, wow, I'm going to lose all of my staff, and I'm going to have a new boss who I don't know if I'm going to like working for them or not, and maybe I should look for something else to do. And so my daughter actually was applying to TJ, and so I had gotten on the website to see when they were going to release those letters to tell you if you've been accepted. And when I did that, I saw this little thing over on the side of the website that said, "Do you want to teach at TJ?" And I thought, and I clicked on it, and that's what happened.
Wow. So what have you learned since becoming a teacher?
Well, I've learned that you can be a scientist whether you are in a lab full-time or not, because I feel like I'm still doing science every day, especially with my seniors who are actually doing research. I feel like learning how to teach effectively is a science in and of itself, and so I try to do experiments with my students to see how it goes, if they enjoy it, if they learn something, if they get interesting results. And you change things up every year, just like you kind of change things up when you're doing an experiment to keep making things better, making improvements. And it's just, I can still do my science just a little bit differently.
So can you tell us a little bit more about some of your favorite past senior research projects that you've seen in the lab?
Well, some of the projects that we have going on have started a couple of years ago, and one of the ones that kind of stands out for me is a project that began as a collaboration with the National Park Service. So there's this black biofilm growing all over the Jefferson Memorial, and actually Mr. Hannum had a connection with someone that he knew who was talking to him about this because she worked for the National Park Service. And he thought, wow, that's a really interesting project. I wonder if TJ kids would like to be involved with that. So we sent out an email to all of the staff at TJ to see if anyone was interested, and I guess I was the only one who answered because I thought, well, that's kind of cool. And I knew that we had this great instrument here, next generation sequencer, that would allow us to identify all the species that were growing on the monument. And so that was how that project began. And we've been working on it for 3 years now. We've gotten some very interesting results that we've shared with the National Park Service. And we've actually moved on to working also at Arlington National Cemetery because they have biofilm problems there. So those projects are a lot of fun because we get to go on field trips to the different locations and collect samples. We actually get to work with people from the National Park Service and Arlington National Cemetery. I've gotten to present student data at a National Park Service meeting and participate in some brainstorming and planning meetings at Arlington National Cemetery about how to get rid of this biofilm, because none of them want it there because it's black and makes everything look very dirty. We've gotten to work with collaborators in Milan, Italy, and from Temple University, who are also studying biofilms, which is pretty cool. And we also have been able to try to grow our own model of biofilms in the lab, which has been interesting, and we keep learning things every year about that. So that's one of my favorite projects. But I also like the cancer projects that we have going on. We have a lot of cell lines that are cancer cell lines, and students come up with interesting ways that they want to try to treat cancer, and I enjoy doing that too.
Cool. So do you have any advice for students looking to go into like the biology related research? What would you like for your students to take with them as they pursue this kind of research in the future?
Well, I think the thing that I would like people to recognize is know something about yourself. Do you have the patience and the perseverance to go into research? Are you comfortable with failure and troubleshooting and trying to do the same thing over and over again until you get it right? Because you really have to be committed to that approach when you're doing science. Science is a hard field. There's a political side, there's a culture that goes along with doing science, and I think a lot of people don't recognize that. They kind of have this glorified image of scientists making breakthrough discoveries in a lab, and they don't realize that in order to get there, you have to have funding, that you have to literally fight for amongst your peers, and oftentimes that's very, very difficult to get. It's difficult to get successful results and get papers published, and it's not an easy path, but it can be very rewarding and very fulfilling, and I think it's important for people to kind of know what they're getting into before they head down that path. But I think once you learn the skills of doing science and being tenacious and not giving up on something, and troubleshooting, those are skills that will last forever, and can be applied to many other aspects of your life.
Cool. Well, thank you so much for being with us here today for the interview, and we really appreciate the stories and the information that you've shared.
Sure, no problem. Thank you for asking.
Thank you for joining us on this week's podcast. Today, we discussed the biological effects of anxiety and stress in the brains of mice. Our guest was Dr. Mary Burnett, TJ's Biotechnology Research Lab Director, who shared with us her own research on stress and experiences as a teacher. Thank you for listening to our podcast, and see you next time on TekTalk.
You were listening to TekTalk.